Okay, so welcome everyone to another Genomics Aotearoa seminar. I hope you're having a lovely day. It is weird weather here in Dunedin. Today we have another fantastic speaker from across the ditch in Australia. So we have distinguished Professor Ian Paulsen, who claims to only work on things that are single cells, so microbiology and microbial genomics. He is the founder and director of the ARC Centre for Excellence in Synthetic Biology, and works on all kinds of things around how to use synthetic biology to develop microbes to do interesting things. I think one of the most interesting things is that he co-founds and directs the Australian Genome Foundry, which I think is a model for some of the things that we perhaps want to do in Genomics Aotearoa. So it's delightful that Ian can join us all the way from Australia somewhere. I didn't get that, but
Macquarie University, Sydney.
Macquarie University, yeah, sorry, from Macquarie University. Ian, thanks very much, and take it away.
Great, thanks, Peter, and thanks for the invitation to talk to you across the water there. And yeah, let me share and get started. So let me start by talking about what synthetic biology is for those who aren't familiar. So it's a relatively newish science. Biology is messy and complex, so to try and make it easy to engineer biology, we bring in engineering concepts like abstraction, modularity, and sort of building standardized parts. And it's got this central conceit that we can essentially treat genes or DNA like little Lego blocks. And we can take a gene from this organism, this Lego block, and a gene from this organism, another Lego block, and put them together in new combinations and design organisms to do things that they don't naturally do. And incidentally, this QR code goes to an animated explainer video that my centre's made on what synthetic biology is. Central to synthetic biology and inspired by engineering is this iterative design, build, test, learn cycle. So the idea then is that we can design our synthetic microbe on a computer and decide what genes we're going to take from where and what promoters and what other things we might need to make whatever it is we're trying to do work. We'll build it in the sense that we'll clone these genes into yeast or into a bacteria or whatever we're trying to do. And then because usually what we're trying to do doesn't work the first time, we'll test it with high throughput omics technologies mostly to sort of see what worked, what didn't work. And then based on that, we'll learn where it went wrong, what the roadblocks are, redesign it, rebuild it, retest it, relearn it, and iterate around this cycle until we've either engineered a microbe to do whatever it is we're trying to do, or we've done enough times we're convinced it's impossible and we'll give up and try something else. And synthetic biology then is this essentially platform technology that has almost an infinite range of applications. A lot of work to date has been focused on trying to engineer microbes to produce biochemicals that we traditionally get from petroleum refining. Can we, engineer microbes to turn, produce biomass into plastic precursors, industrial platform biochemicals, things like that. But there's also diverse other applications in biomedical sphere, improving agriculture, bioremediation, producing fuels and producing novel biomaterials. I'm director of the Australian Research Council Centre of Excellence in Synthetic Biology, which is essentially Australia's flagship program in synthetic biology. So Centres of Excellence are 7-year have a 7-year runway. Our centre has received $35 million from the Australian Research Council and raised an additional $15 million from our industry and academic partners. So it's a 7-year, $50 million research centre. We're about 4 years in at the moment. In fact, in a few days' time, we probably hit the 4-year mark. And broadly speaking, our vision is, can we take either sustainably produced biomass or waste streams such as industrial gases or agricultural or municipal wastes and engineer microbes to turn those into high-value products, pharmaceuticals, platform biochemicals, fuels, biomaterials. And in theory, this QR code should go to explain a video about what our centre is, but I actually think it's the wrong one that's going to the same, and it's the same as the other QR code on my previous slide. And our research program is divided into 3 themes and 3 capabilities. So our 3 themes are essentially engineering microbes at different levels. So we're trying to build synthetic microbial communities where we can get microbes to work together to do things that no one organism could do by themselves. We're trying to engineer synthetic organelles, repurpose eukaryotic organelles like the mitochondria to sort of act like tiny little bioreactors for production of very high-value compounds within a cell. And in neobiochemistry, we're trying to build entirely new to nature enzymes or pathways to enable biochemical transformations that no natural microbe can do. And then our capability systems, bioengineering, is essentially the automated design, build, test, learn cycle that we use to undertake our synthetic biology, or to accelerate our synthetic biology research program. Industrial translation. This isn't a team doing actual translation themselves. They're actually a team of engineers and humanities and social scientists and economists looking at what are the factors, what are the sort of government policies and regulation and industry strategies that might either enhance or hinder the uptake of our synthetic biology technologies, particularly in Australia. And then in social dimensions, we have a team of humanities and social sciences, researchers with expertise in ethics, in technology law, in sociology and anthropology, so sort of exploring the policies and practices that surround synthetic biology. And so we're this large, diverse centre. We're essentially like a giant octopus with sort of a tentacle everywhere. Currently, the centre it's led out of Macquarie University in Sydney, but it spans 9 universities, 17 research groups, and we have 30 industry partners or affiliates. So in total, we have about 250 researchers across the centre, and this is a photo of one of our probably about 180 to 200 of our centre members at one of our centre annual conferences. So when the Australian Research Council funds a Centre of Excellence, you're funded not just to do cool research, but they hope that you're going to have an impact in the real world. So one of the things I'm particularly excited about is that within our first 4 years, the Centre has spun out 9 startup companies that have now collectively raised more than $190 million of venture capital. I'm not going to walk through all of them, just give a couple of examples. So Samsara Eco, spun out of Colin Jackson's group from the Australian National University, and they have engineered enzymes for breaking plastics down to their monomers. And so they've just raised money to build their first full-scale plant. They actually don't use organisms. They've purified the enzymes and put them on a solid matrix and then run a plastic slurry across them. But their first plant is taking PET plastics from plastic drink bottles and recycling that back to the virgin monomers that can then be used to build new plastics afresh. And Number 8 Bio, spun out of my group by one of my postdocs and one of my PhD students. They are engineering yeast strains to produce anti-methanogenic compounds, and those yeast strains can then be incorporated into cattle feed and stop cattle producing methane. And they've now done several cattle trials with various product formulations and shown 95% methane mitigation with their lead products. And with their most recent product, also see an increase in animal productivity, as in the animals gain more weight on the same amount of feed. So instead of carbon energy being spent making methane. It's now going into making cow biomass instead. Where a Centre of Excellence is also supposed to have other impacts in the real world, and one of the things I'm a big gaming nerd, and I'm particularly excited that the young researchers in my centre have developed a educational card game, which, having played alpha and beta tests with it myself, I can tell you it's actually a fun game to play. We've run public events and events at schools around Australia with this game. It's now undergoing. They now have a final beta test version, about to come up with a final version they hope to take into commercial production. They've also been working with schools in New South Wales to develop a curriculum around it, so that actually a school curriculum going to be spread to schools around New South Wales using this card game as an educational tool, and we hope that can then be used more broadly throughout the country. And in this game, you engineer. You're basically you play a microbe and you put new genes in it to give yourself new capabilities and try and survive different environments and try and remediate plastic out of the environment. Based on those microbial characters, we're also making a range of other outreach materials, such as children's books and games for younger children. The other entity I'm the director of, and this is I think probably of more direct interest to Genomics Aotearoa, is I'm also director of the Australian Genome Foundry. So the Australian Genome Foundry is a high throughput synthetic biology facility funded by the Australian equivalent of Genomics Aotearoa, Bioplatforms Australia, with support from NCRIS, the National Cooperative Research Infrastructure Strategy for Australia. And so the idea of this facility is that we're essentially trying to bring synthetic biology into and molecular biology into an industrial era. So into an industrial revolution sort of era. So traditionally, molecular biology has sort of been a cottage industry. You've got PhD students and postdocs, essentially skilled artisans with years upon years of training, sitting at a bench doing repetitive tasks over and over again. And so the idea of a genome foundry is that we can use robots to do those repetitive tasks and free people up to do more creative things like do the designs and interpret the data, rather than spending their time doing mundane, repetitive work at a bench. So the videos here are just showing colony picking and liquid handling robots doing their stuff. This is our labs at Macquarie, synthetic biology labs at Macquarie. So over here is actually the Centre of Excellence labs. Here is a space companies it can incubate in, and these are the robotics of and analytics of the Australian Genome Foundry, and it goes on further to the right out of shot. And so to date, Bioplatforms Australia has funded 2 facilities, the Australian Genome Foundry at Macquarie, and a second facility, Idea Bio, led by Esteban Marcellin, who's a member of my Centre of Excellence, but also leads Idea Bio. And these are sister facilities providing complementary capabilities. And so at the Australian Genome Foundry, we can do high throughput strain engineering. So if you want to clone a few genes or knock out one gene, you should just get your PhD student or postdoc, whatever, to do that. But if you want to make strains where you're making 100 strains or 1,000 strains, or you want to make, clone a particular, genes for a particular pathway, but you want to make 300 different variants with taking genes from different organisms, then instead of having a student try and do that, spend years upon years trying to do that, you should come and talk to us and we can, do high throughput robotic-based strain engineering. We also do fermentation, but if you want to grow things in 1,000-litre tanks, we don't do that. I don't know anything about that. We do high throughput fermentation. So we can grow dozens or up to about 50 strains in parallel under industrial relevant fermentation conditions to see how well they perform at that level. And then we have a very nice analytical chemistry suite with GSMS, LCMS, HPLC, but these are set up not to do shotgun metabolomics. If you want to do shotgun metabolomics, there's other facilities funded by Bioplatforms Australia that do that. We're set up to do high throughput targeted metabolomics. So there's no point being able to build hundreds of strains if you can't test hundreds of strains. So our analytical chemistry suite is built to test hundreds of strains in parallel, but looking to identify and quantitate particular individual metabolites. That what have you tried? Usually if you try and engineer your microbe to produce compound X, then we can test for compound X and its various precursors. And then lastly, we have a flow cytometry, flow sorting suite set up specifically to do microbial flow. The other thing that the AGF does that is, I think, unique amongst biofoundries in the world is we are set up to allow startup companies to incubate in the biofoundry. So I currently have 4 companies incubating in the biofoundry. Some are startups out of Macquarie, some are companies that have relocated to Sydney in order to get access to the bio capabilities of the biofoundry. So these companies pay an annual fee to directly access, to embed staff in our facility, and they can directly access all the infrastructure in the foundry. In terms of how the foundry operates, we're set up to try and be as flexible and nimble as we can. So when you're servicing clients such as startup companies who have very short timelines that are not the timelines universities traditionally work on, we've arranged to do an end run around most of the university bureaucracy in order to be able to respond to our clients. So Australian universities are, flexible and nimble are not adjectives that are traditionally associated with Australian universities. So we've sort of worked with the university's streamlined system to enable us to do that. And we essentially have 3 business models or 3 modes of interaction with clients. So first of all, we can do simple fee-for-service work. Client comes to us, wants some strains built or tested or whatever. We can do the work. They pay us a sum of money. We give them the data or the physical materials or whatever. 100% of the IP sits with the client. With the startup model, companies pay an annual fee to access the facility, to be embedded in the facility and use all the approximately $12 million of infrastructure that we have in the facility. And again, they own 100% of the IP. There's no IP reach through. And then lastly, we can do more collaborative projects where someone comes and says, we want a strain that does this. We don't know how to build it. Please make it for us. In that case, IP would be shared on some mutually agreed model. And the other thing to mention about the Australian Genome Foundry, because we're funded by NCRIS and by Platforms Australia, we don't have to charge full cost recovery. So the salaries of our staff are largely paid by money from Bioplatforms Australia, and so we can provide extremely cost competitive services in these spaces because we don't have that requirement for full cost recovery. Our sister facility, Idea Bio, basically is set up to do, whereas we're set up to build hundreds of strains, they're set up to test a small handful of strains. And so they do larger scale fermentation using 5-litre bioreactors. They have a very nice suite of multi-omic capabilities, and then are set up to do computational modelling. So the vision is, and we have a number of joint projects, that we can do initial designs here, build, let's say, 100 strain variants, do high throughput testing to see which of those are best. We can, take the 5 best strains and send them up to Idea Bio. They can then do a multi-omics, do computational modelling, and then come back to us and say, you should redesign your problems are here and here. You should redesign your strains like this, and we can then iterate around the synthetic biology design, build, test, learn cycle. And so in terms of where the Centre of Excellence and the Genome Foundries sit in the Australian, what I see as the emerging Australian bioeconomy ecosystem, the Centre of Excellence is at this end, and if this is the pathway of a startup company, then the cool science that might lead to ideation of new companies is being done at the Centre of Excellence or in other synthetic biology labs or facilities in Australia. Companies can then go and do some sort of startup accelerator program. The ones that Australian SynBio startups tend to do is the UNSW SynBio 10X Accelerator or the CSIRO on Accelerate on Prime program. And then where the biofoundries fit in this ecosystem is we can help companies prove at laboratory scale that what they're trying to do works, and we can accelerate their journey. So, for instance, the founder of a number at Bio is on record as saying the only reason they were able to reach their targets that enabled them to get their most recent round of funding is because by being embedded in the foundry and having access to the high throughput foundry equipment, they could, achieve their research goals much more rapidly than if they were in any other environment. And then there's various commercial and academic facilities that can do pilot scale fermentation up to 1,000 to 10,000 litre scale. For the commercial ones include Cauldron and FTech / the Hunter Biological Solutions Facility. But there's also academic options within the Centre of Excellence where there's the 2,000 litre Mackay pilot plant. And then this is where we get more in my imagination, where we don't really have companies yet doing production scale in their own facilities in Australia, although Cauldron and Samsara are the two that are probably closest. All right, so I've been talking very high level. Let's talk about some actual research stories. And so I got into synthetic biology 10 years ago. I was originally a genomics and metagenomics and systems biology sort of person. We still do all that stuff, but now synthetic biology is a large part of my focus. And we sort of, instead of dipping our toe into the synthetic biology water, we sort of threw ourselves into the deep end of the swimming pool. And so we actually joined the Yeast 2.0 Global Consortium, which is a group of laboratories across 6 countries building the world's first synthetic eukaryote. What does that actually mean? We've redesigned the yeast genome. We've chemically synthesized DNA to build those individual chromosomes. We've built synthetic chromosomes replacing the natural chromosomes. And each lab or each team within the consortium takes responsibility for design and building of one or more individual chromosomes. We were actually the last team to join, so we got the 2 chromosomes that no one else wanted, which were chromosomes 4. Where's my mouse going? There we go. Chromosomes 14 and 16, and I now know why no one else wanted them. We're all working to a common design template. So in terms of these synthetic chromosomes, we've replaced the natural telomeres with synthetic telomeres. We've removed all retrotransposons and other repeat regions in the chromosome. We've changed all of the TAG stop codons to TAA. So that frees up a codon that could be used in the future for incorporating unnatural amino acids in the protein. The original plan was to delete all of the introns in yeast, but that's proven not to be possible because one of the accidental discoveries of the project is how many introns in yeast are actually essential for yeast function. And then throughout the yeast genome, we've built in LOX-P sites for the system called Scramble. So these are site-specific recombination sites. And that means we can turn, with the addition of a simple chemical trigger, we can turn on very high rates of genome rearrangements, deletions, insertions, inversions, and duplications. And the end result of this is about, the genomes tend to be somewhere on, but depending on the particular chromosomes, they're either 10 to 20% smaller than their natural chromosomes. So how do you actually build a synthetic chromosome? First, we design it on the computer, and it's designed in 5 to 10 kb pieces we call chunks. And these are built by external commercial providers such as Twist or GenScript. We then take that chemically synthesized DNA, and we usually take 4 or 5 chunks and ligate them together to make what we call a mega chunk. And then we'll transform that into yeast, and we'll select for a marker in our particular mega chunk, and we'll override a marker on a previous mega chunk. And we can literally walk across each chromosome, replacing 40 kb at a time. Now, we started this project in 2014, and we published chromosome 14, I think, last year, and we have chromosome 16 essentially accepted in Nature Communications. At the moment, we need to do one last change to one figure, and then it's accepted. So this was a 10-year project. The thing is, we can build a chromosome in 6 months. We don't need 10 years to build a chromosome. So why did this project take 10 years? It's because as we walk across that chromosome, replacing 40 kb at a time, one of the design goals of Yeast 2.0 is at the end we should have a synthetic yeast that has wild type yeast fitness. So we don't want to make a sick, useless yeast. We want a synthetic yeast that is still fit. And what we find is maybe a quarter, a third of the time, when we put in one of our synthetic 40 kb mega chunks, we cause some massive loss of fitness from one of our changes. And debugging what changes we've made that cause that loss of fitness has been tremendously time-consuming and annoying. And so we can build a mega-base size synthetic chromosome in 6 months or less. For chromosome 16, it's taken us 10 years to debug the fitness defects of accidentally incorporated, and these are just some. I'm not going to go through them all. These are just the various fitness defects we fixed in chromosome 14, but just to give one example. So MRPL19 is an important mitochondrial ribosomal protein. The design incorporated a LOXP site at the end of that gene in the 3' UTR region, and what we found is that this caused a massive loss of fitness and essentially loss of the mitochondria. And after a lot of debugging, we worked out our first thought was that it was affecting mRNA stability, but that's not actually. It doesn't affect mRNA stability or expression. What it actually affects is targeting to the mitochondria. And so when we made fusions to GFP, and here's free GFP just being expressed, and of course it goes to the cytoplasm, and here's a mitotracker dye showing where the yeast mitochondria are. In the strain where this LOXP site is incorporated into the 3' UTR of that mRNA, the fusion protein is not being targeted to the mitochondria. You can see here it's going somewhere else. And when we remove that LOXP site to our synthetic MRPL19 gene, it's now being targeted correctly. There's my mouse going. There it is. So we can rapidly build synthetic megabase size chromosomes, but to debug them when we redesign a natural one is a painful and time-consuming process. So we're interested to see, can we build entirely new to nature chromosomes? And our first attempt to do this was in collaboration with Anthony Borman's group at the Australian Research Institute. And so Anthony's team had done sequencing of a range of industrial yeast strains, from winemaking, from baking, from beer brewing, and from ethanol production. And they had identified a whole range of genetic loci that were present in these industrial strains, but missing in the lab strain. And certainly the standard yeast lab strain, BY4741, is not something you'd ever want to make wine or bread or beer with. And so we built, it's a bit of a mouthful to say, we built a pan-genome neochromosome. So we built a new chromosome, took all of these genes that we'd identified in these industrial strains and built a new chromosome that we put into our standard lab strain. And what we found is that actually increased the robustness of the strain for these sort of industrial processes, and it also, interestingly, enhanced the ability, or actually gave the lab strain the ability to use a range of sugars that it couldn't normally use, but which the industrial strains could use. And the interesting thing about this is that this strain was basically, this synthetic neochromosome was built in a few months, and it didn't cause any significant, there was no significant fitness loss. So it sort of taught us that redesigning an existing chromosome is actually a great pain in the ass, but to build a new to nature, entirely new chromosome in a particular strain that doesn't have those genes actually seems to be much easier. And so I had a PhD student, Tom Collier, who he and his wife are very passionate about deforestation in Southeast Asia. One of the drivers of that is building plantations for the production of palm oil. And so Tom wanted to see, could he engineer yeast strains to produce palm oil alternatives, and also alternatives for other vegetable oils. And so rather than doing it, and lots of people have tried to do, like Jens Nielsen's group has done a lot of irrational engineering to try and do this. Tom wanted to see, could he build a new to nature and neochromosome aimed at producing triacylglycerides in yeast. And so Aotearoa lipolytica is another yeast which is famous for being an oily yeast, for producing a lot of lipids. So Tom took every gene we could conceivably think of that was relevant to production of yeast lipids, production of biosynthesis genes for making triacylglycerides, lipid droplet proteins, and proteins for making precursors and improving, reducing power and supply of precursor chemicals, and designed a neochromosome with those genes from Aotearoa lipolytica, and then built that in Saccharomyces cerevisiae. And he built this in 16 individual chunks, where each chunk was focused on the genes for some particular part of those systems. And this is just doing a Nile red stain is a quick and dirty test for production of triacylglyceride production. And with each of those individual chunks, none of them really did much, with the exception of chunk 13, with some extra glycolysis and glucose transport related genes had a somewhat increased staining of Nile red. He then, because by this stage was getting towards the end of his PhD, he tried a quick and dirty, I'm going to try and build a synthetic neochromosome in a one-pot assembly by doing homology-directed repair with these 16 chunks. And he wound up building essentially a library of neochromosome variants, probably none of which are actually totally complete, but which have various combinations of those 16 chunks. And this is then just doing a Nile red stain. And these are those two bar graphs from, this is the wild type and this is our best. Actually, no, sorry, these two different wild type strains, a standard lab strain and a sort of more industrial strain. And this is their Nile red staining here. And what you can see is we had a range of variants producing a crap load more triacylglycerides or lipids as suggested by Quickandini-Nile red staining. We then did lipidomics for production of total lipids and individual tags, and also individual triacylglyceride production, and other individual lipids. And what we found is that we've produced at least an order of magnitude more triacylglycerides and total lipids with this, and we think this strain is the highest producing yeast strain ever made for triacylglyceride production. Unfortunately, in retrospect, we made naive design decisions, and this chromosome is not very stable, and so Tom has actually spun out a company, Lever, to try and commercialise this, but actually they really need to rebuild this from scratch because the strain is not really stable enough for industrial fermentation. Okay, I'm going to change from building synthetic chromosomes to talking about engineering synthetic symbioses. So, of course, symbioses have been very important in the evolution of life on Earth. For instance, key in the evolution of eukaryotes in terms of mitochondria coming from an ancestral endosymbiosis of an archaea and a bacterium, and the evolution of plants with chloroplasts having derived from the ancestral endosymbiosis of a cyanobacteria. And so this sort of falls into the building synthetic microbial communities arm of our centre. And so Aotearoa led this along with and worked with another postdoc, Steffi. And so they engineered a split protein system. So hygromycin D is an antibiotic which will kill both eukaryotes and prokaryotes. Hygromycin D phosphotransferase is a resistance protein. They designed a split protein system where you could express half of it in one cell and half of it in a different cell, and then the two organisms would only survive in the presence of the antibiotic when both of them were there. And I engineered this system and initially put it into yeast. So here's some microfluidic wells with cells expressing this half of the protein are also producing a red fluorescent protein, M3, I think, and the cells expressing this other half of the split protein system are expressing GFP. And if you have cells just expressing one half of the protein, they don't grow. If you have cells expressing the other half of the protein, they don't grow. If you put them together, then the two of them grow together and can survive in the presence of the antibiotic. In this particular system, the split proteins are being expressed on the surface of these two yeast cells. And what we've found is this system works when we're doing it in microfluidics, but when we scaled up even to like 10 mil, it doesn't work very well. So this is, we then actually before I talk about what we did, then let me talk about how we designed it. So we actually made 4 different splits, and what we found is that one of them worked a little bit, and then in order to make that improve that to not quite as good as a non-split, but almost as good as a non-split, we actually put in a protein docking system to more effectively bring those two halves of those proteins together. We then decided what we wanted to do was to try and build a synthetic photoendosymbiont. So we wanted to express one half of the split protein system in a eukaryote, in this case the amoeba Dictyostelium, and the other half in a cyanobacterium, in this case Seneca cystis. And in the case of the cyanobacterium, express on the surface of the cyanobacterium, and in essence try and recapitulate the ancestral evolution of the chloroplast by forcing a cyanobacteria to live inside a eukaryote. One advantage of this system is that cyanobacteria are photosynthetic. They can make chlorophyll, So we can easily monitor cyanobacteria internalization by confocal microscopy, looking at the fluorescent pigments of the cyanobacteria, looking at chlorophyll. The other advantage the system has is dictyostelium. When it's live, it's amoeboid and it's motile. When it's dead, it's non-motile and it becomes spherical. And so then we can use confocal microscopy to monitor whether the system works. So here's our positive control. Here's a dictyostelium. In this case, we're expressing a full-length hygromycin, the phosphotransferase, in the dictyostelium as a control. So in the presence of the antibiotic, the dictyostelium should live. And so you can see the cells are amoeboid and sort of motile. And that's regardless as to whether they've got a cyanobacteria internalized or not. So that's a positive control. Here's our negative control. We've got the dictyostelium expressing one half of the split protein system and the cyanobacteria expressing the same half. So they're not coming together to form a functioning thing. So here's our spherical non-motile dictyostelium dead, and it's dead regardless as to whether it's got the internalized cyanobacteria or not. And then we were super excited when we saw this video. So here's our split protein system in reality. So we've got one half of the protein being produced on the surface of the cyanobacteria, and the other half of the resistance protein being produced in the dictyostelium. So when dicty doesn't have cyanobacteria internalized, it's spherical, it's non-motile, it's dead. And when it's got the cyanobacteria internalized, the dictyostelium is amoeboid and motile. And we have tested now this system up to, I think, 1 to 2 weeks, and the dictyostelium and the cyanobacteria survive in the presence of antibiotic over that time frame. However, the system has its issues. One issue is, as far as we can tell, the dictyostelium isn't able to divide, and we don't quite know. This is data that's still sort of hot off the press, so we're still working out what's going on. We do see dictyostelium cells that have multiple cyanobacteria in it. We don't know if that's because they've engulfed multiple cyanobacteria or if it's because those cyanobacteria are able to replicate in the system. We have about 100 different design variants that were built using the robotics and the Foundry that we're exploring to see how they perform. Rather than doing it all by confocal microscopy, we've now worked up a flows sorting method and flow cytometry method to count and monitor the performance of our synthetic endosymbionts. What we do know is that maybe only somewhere between 1 to 5% of the dictyostelium successfully take up the cyanobacteria and survive. We don't know if that's we don't know if that's just the rate of transformation we're getting or the rate of engulfment, or whether that's or whether there's something else that needs that there's a reason it only occurs 1 to 5% of the time. So we're still trying to optimize and perfect the system. We can now use flow cytometry to enrich for our populations of our photoendosymbionts. So we're hoping that will fast track our ability to explore things with this system. So what we'd like to do is we can only study endosymbiosis now by sort of looking back at what, using genomics to look at what happened, hundreds of millions of years or years ago. So by using this system, we're hoping to be able to use omics and do experiments over time to understand how organisms adapt to each other when they're in a new recently established endosymbiosis. And then, talk very quickly, we're also doing other work to try and build synthetic microbial communities. C. necator is an interesting organism that can grow on CO2 or formate. And so we're trying to engineer and Paige in my group is trying to engineer a synthetic syntrophy between a yeast and a bacteria. So the idea is that we can get C. necator to grow on CO2 or sodium formate, to make sucrose, to secrete that sucrose, which we've then engineered a sucrose transporter and knocked out the signal peptide in the native sucrose invertase gene, and we can get that yeast to overproduce tryptophan and secrete that, which we then hope to be able to make a tryptophan auxotroph and C. necator that could cross-feed. And so we've done all the engineering in yeast that we need to do, and we've engineered C. necator to make the sucrose, and we've shown that we can cross-feed Saccharomyces with the sucrose from C. necator. We have not yet made a tryptophan auxotroph and completed it. So that's work that's ongoing but under development. And in the longer term, we'd actually like to make a three-way syntrophy between C. necator and a nitrogen-fixing bacterium, Azotobacteria, and yeast, with the idea that C. necator can fix CO2, Azotobacter can fix nitrogen. Those can provide, Azotobacter can secrete ammonia, and C. necator can secrete sucrose to feed the yeast strain, and the yeast can then cross-feed back and complement amino acid autotrophies in those two bacteria. And we can essentially build a three-way system that could then produce high-value compounds from CO2 and nitrogen. And so I've given you an overview of the ARC Centre of Excellence in Synthetic Biology and the Australian Genome Foundry. And then on the sciencey side of things, we've engineered 2 synthetic chromosomes as part of the Yeast 2.0 project. And we've now built 2 neochromosomes, one of them a lipid neochromosome for high production of triacylglycerides. And we've engineered a split protein system to build synthetic symbioses between eukaryotes and prokaryotes. And we're working on other split protein systems and also working on trying to develop synthetic syntrophies between yeast and bacteria. And I think I'm out of time, so I won't. That's something else I was going to talk about if I have time, but I don't. So this is my team at Macquarie and the various funding agencies that have supported us, and that's it from me.